Tonight we have Maria Mody, who's joining us. She is an assistant professor of radiology, and the research that Maria generally is involved in focuses on neurocognitive architecture of language, attention, and reading processes, both in typically developing individuals and in atypical development, like we see in autism. Her specific research interests include dyslexia, autism, and specific language impairment, and she has been working with us for a number of years on some research initiatives related to our minimally verbal population of individuals with autism. So we're so excited to have Maria here. Joining Maria, we have Evan Bordt, who's an instructor at Harvard Medical School. And Evan is one of our basic science researchers. He studies neuroimmune interactions and neuroimmunometabolism in brain development with a focus on understanding sex differences in neural function in order to better understand how these processes influence things like social behavior and communication. And certainly we can think of lots of ways that Evan's research really directly applies to human conditions like autism. So we are actually going to kick things off with Evan. I'll pass the floor to Evan here to give us a little talk and tell us a bit about his research. As Evan and Maria are talking this evening, please feel free to use the chat function to send some questions our way. I will be facilitating a question and answer period after each of our presenters has an opportunity to tell you briefly about the work that they're doing. So with that, I'll pass it to Evan to kick us off.
Thank you so much, Lisa. Just want to make sure that you guys can all see my screen. All right. So yeah, thanks so much for the opportunity to present some of our preclinical basic science research this evening. And this evening, I really wanted to kick this off and discuss just some of the ways that we can assess communication in rodent models of autism. So we all know that autism is characterized by hallmark behaviors such as alterations in social behavior, communication, as well as some disabilities with language. And much of what we have learned about autism to this point mechanistically has really stemmed from the use of animal models. One such animal model that has proved particularly useful to us is the use of rodents such as rats and mice. And rodents are very useful for understanding the mechanisms that really can lead to or exacerbate different aspects of autism, because they provide us with the ability to manipulate certain things, such as specific genes in a specific cell type at a different time. And they also give us the ability to dissect tissues or cells that would not necessarily be accessible in a human. Just for instance, in a mouse, we're able to dissect out particular types of brain cells from an actual brain, and that's something that we would not be able to do, obviously, in a living human. So there's a lot that we can understand from these rodent models of autism, but at the same time, I think it's very important to remember that there is absolutely no such thing as a mouse with autism. It's simply a model that we're able to use to really understand the underlying biology. But for the purposes of today's discussion, we're really interested in how rodents are able to communicate with each other. This video that I am showing here is a common task that's used in laboratories. It's called the three-chambered social interaction task, in which our mouse of interest is placed into a three-chambered arena and allowed to investigate the arena itself or any number of objects that are contained within these little plastic cups on each side. In this case, our test mouse is allowed to interact with either a rubber duck, which we see here on the right, or with a novel unknown mouse on the left. And we can see here that these mice are clearly interacting with each other. And we know that one major way that mice can interact with each other is through their sense of smell. And indeed, smells are a hugely important route through which mice and other rodents can get their point across to others. However, we also know that mice are actually able to communicate with each other in a way that is somewhat similar to our human verbal communication. But you might be thinking that you've seen mice, either out in a field or annoyingly running around through your house sometimes, and you can't really hear much of what they would be saying to one another past an occasional squeak or something similar. And much of this actually has to do with limitations in what humans can hear. And as you can see in this graphic, humans can hear between the frequencies of 20 hertz and 20,000 hertz. And that's actually a really small frequency of sound. I think this is best visualized by the fact that if you have a dog or a cat as a pet, you can tell that they often hear things that we're just not able to, and that they're reacting to other sounds or creatures, such as bats can actually voice a lot of what bats say at ultrasonic frequencies that we're not able to hear. And if you're outside at night with your dog, you can see, and you see a bat, you can tell that they're reacting to something that we might not be able to hear. And it's pretty obvious that there's just a whole lot of sounds out there that we're not capable of observing using just our natural hearing. And this is highly relevant to studying rodent communication, as a really landmark study nearly 70 years ago actually first observed that lab rats are capable of producing sounds in the ultrasonic, or very far to the right of this graph, frequency. And indeed, recent studies have determined that the majority of rodent vocalizations actually occur in the range from 30,000 to 110,000 hertz, which, as you can see here, is well past and into the ultrasonic frequency that we just can't hear. And this means that we need some type of specialized equipment beyond just normal microphones to really assess rodent communication in a rigorous manner. So how can we do this? So for the most basic of these experiments, we're able to use these specialized microphones, like the one pictured in the top left, that are able to capture in the frequency of 2,000 to 200,000 hertz. And remember that humans can really only hear up to about 20,000. So this allows us to capture a lot that we could not with a normal microphone or with our ears. And what we can do is we can place these ultrasonic microphones above a container that has our mouse in it, and place this inside of a sound attenuating chamber, such as the one we see here. And this is essentially what we use. It's just a large box that's fully covered inside with a sound dampening foam. And then we hook the system up to a specialized receiver in order to record what our rodents are vocalizing. And if you notice, these basic experiments use only one mouse inside of a sound attenuating chamber. So how are we able to get this single mouse to say anything when it's by itself? So others have discovered that infant mice actually emit these ultrasonic vocalizations when they're separated from their mother. And as you can see in this video, when infant mice are separated from their moms hear these vocalizations, which are essentially a, hey, come and get me signal. And we can see here that mother mice are actually very responsive to these calls, and they actively retrieve their young and bring them to safety. So taking advantage of this knowledge, we can place infant mice in these sound attenuating chambers away from their mothers and record their ultrasonic come and get me vocalizations. So we attach these microphones to our specialized receiver and use software to record these vocalizations. And then members of our team can use specialized software such as this mouse ultrasonic profile extraction or Muppet analyses to really make sense of all of this data that we've recorded. And I'm going to try to play you a sound right now. Can someone just, I was struggling with this earlier today, so can someone make sure that you can actually hear the sound when I stop talking in a few seconds? But what do these USVs or ultrasonic vocalizations really sound like? So what we're seeing here is essentially a spectrogram or visual representation that's generated from the input of our ultrasonic microphone. And like I said, I'm hoping that once I click play on the sound here, that you're able to kind of hear along with the visuals of the spectrogram of about 25 seconds of an infant mouse calling out for its mother. And I'll try to follow along with my mouse on the spectrogram as we hear what it is that this mouse is saying, transposed into a frequency that our ears are actually able to distinguish. So, as I'm sure you can tell, these are actually quite complex vocalizations that these little mice are actually making. And we're able to analyze many different aspects of these calls, from more basic aspects such as how often are they calling to far more complex levels of analyses. And just as an example of a really basic analysis, this is some data from a genetic mouse model of ASD. And what we can see here is the number of vocalizations in a control mouse compared to the number of vocalizations in this ASD mouse model. And we can see that the ASD mice, this particular genetic model, have dramatically lower or fewer vocalizations than do control mice. And a large focus of research here at the Lurie Center is stemming on defining an immune subtype of ASD. And as such, we are regularly assessing vocalizations in mice who were just exposed to various immune activating challenges. And just as an example, what I'm going to show here is that we assess vocalizations in mice whose mothers were exposed to a virus during pregnancy. So if you remember that 3-chambered social behavior task video that I showed you a little bit earlier, we can get a number that's called sociability, which is essentially the percentage of time that our test mouse is engaging in a pro-social behavior. And we're actually then able to correlate this with how many vocalizations these mice make in our early ultrasonic vocalization task. And we think this is really exciting because we can see that there's actually a strong correlation between the rates of vocalizations, which you see here on this x-axis, and the social behavior of individual mice, which we see here on the y-axis. And what this kind of means is that we can actually predict, based off of how the mice vocalize earlier in life, how social they're going to be in these later early adolescent sociability assays. But I think something that might be even more intriguing is we find that this maternal immune activation with this virus, this IMQ virus, actually disrupts this correlation in male offspring, suggesting that something about immune challenges or in these ASD models are really disrupting this social communication network. So to this point, we really only shown some simple data such as, the total number of calls that are emitted during one of these recording sessions. But in order to more deeply understand the complexity of these vocalizations, about 15 years ago, Jacqueline Crawley's lab pioneered a deeper investigation of how these vocalizations can actually establish categories of syllables exhibited in their calls. And the full set of syllables that a mouse produces, so each of those little squeaks that we heard earlier, is referred to as their syllable repertoire. And while we don't yet understand the meaning of any of these individual syllables that a rodent may produce, we've progressed in our ability to categorize these different types of emissions or different types of calls. And in doing so, we've actually been able to essentially categorize 10 different types of syllables that mice emit. And you can see these 10 different types here. And while syllable complexity, of course, exists on a spectrum, bins such as, complex and harmonic live on a more extreme end of the spectrum, whereas short or upward or downward are living on these less extreme ends. And one way that we can really computationally analyze what these syllables are like is to assess how similar they are. Now, this is an absolutely crazy graph to look at. We don't need to really get much out of it. But essentially what this is showing is a correlation between different types of bins of these syllable structures. So essentially a little red square here means that it's exactly the same, and a little dark blue square here means that the structures are quite different. So if syllable repertoires are generally similar between animals, so if two animals sound very alike in terms of how they vocalize, you would expect to see a lot of red in these graphs. And as you can see on the left here, when comparing essentially one of our control male mice, the repertoire of this mouse to the average repertoire of all of our control mice, there's not a lot of this dark blue. And in fact, this black box, which can indicate areas of dissimilarity, so calls that are not similar to each other at all, we can see is quite small. However, when we look at the immune challenged male mice on the right, we can see that there's far less of this red similarity, and indeed a much larger box of this blue dissimilarity, which is really suggesting that the syllable repertoire of what our mice are vocalizing is quite distinct. And well, I just want to emphasize again, we obviously have no idea what these different calls mean exactly. It's also quite obvious that in our ASD models, there's a significant change in how these mice are vocalizing. And this is just one other way of presenting this repertoire similarity, this time in a model of environmental pollution and stress that is shown to induce social behavioral alterations. And as we can see on this right graph here, there's a significant increase in the number of these less complex, kind of shorter calls in mice that were exposed to this environmental challenge than there were in control mice, who tended to kind of have longer syllable vocalizations. Now, this graph is just insane. You don't really need to get anything out of it, but everything that I've really presented today has involved individual mice recorded inside of a sound attenuating chamber. We can tell this is an incredibly artificial environment that never happens in real life. So while it's really useful, there are, of course, far more relevant tasks in which we can record vocalizations. And perhaps the most useful one of which would just be to allow mice to interact in an open arena with each other. And of course, when you think about this, using a traditional single microphone technique, this has proven nearly impossible because we've been unable to tell which sounds were really coming from which mouse. However, using these kind of multi-microphone arrays and cameras, last year, researchers were really able to start to be able to determine which mouse is emitting which vocalization at certain times. And this type of analysis, well, undoubtedly it's going to be, very expensive and quite complex, will really be necessary for allowing us to truly determine what kind of relevant alterations are occurring in mouse communication. And I think it's very clear that this type of method that will be used across rodent research to really understand vocalization behaviors in the next few years. So just a quick, I think, three-point summary of kind of what we discussed today is that mice can emit vocalizations in ultrasonic frequencies that we're not naturally able to hear, that various mouse models of autism all have been shown to impact rodent ultrasonic vocalizations. And moving forward, there's just a really, really strong need to move these analyses into more complex and relevant paradigms to really understand differences in vocalizations, which I think Maria is going to get some really great human data that I think will kind of put some of this into context of what we really need to be able to understand on a mechanistic level and why we need to understand what exactly it means when a mouse is vocalizing even better. So I just wanted to thank all the amazing members of my lab and at the Lurie Center who have essentially done all of the work that I showed you today.
Thank you, Evan, for giving us such a great overview of the work that you and your team are doing. As I popped into the chat and said at the beginning, please feel free to start thinking of your questions, especially those that are relevant to Evan's talk now. You can send those to me in the chat and I'll collect those. I've got a few trickling in, but think about other things that you're wondering about while we pass the floor to Maria for her presentation tonight. We'll get you unmuted here and then. Looks great, Maria. Thank you.
All right. So thanks very much, Lisa, for the invitation to Lurie Center. And thank you, Evan, for an amazing platform you've set up for me to just dive right into and continue with. So the focus of what I'm going to do is pretty much building exactly on what Evan has been talking about, which is the whole concept of vocalizations. And unlike the vocalizations that Evan spoke about, which are these ultrasonic sounds. Let me just move. For some reason, there we go. this work that we're going to talk about, the sounds that we've collected from all these individuals with autism and our analysis would just not have been possible without a large-scale collaborative effort. So right here we have at the Lurie Center, a whole bunch of folks led by Chris McDougall, and our collaborators at MIT, Lincoln Laboratory led by Tom Cattieri and his group, and a subgroup at the Media Lab, which led by Patty Mace and her group. And none of this work obviously would be possible without the funding that we have from the Nancy Lurie Marks Family Foundation and the Simons Foundation. So diving right into the act of speech. I just want to make sure, by the way, is my screen visible? Everything look okay? Thank you. So the way I like to think about speech is that it could be divided under two heads. One is the physical act of speaking, and hence I call it a motor act. The output is sound or vocalization. And you can also think of speech as a linguistic act. And importantly, because just the act of making a sound from our mouth doesn't necessarily constitute speech. There has to be communicative intent. There has to be meaning attached to it. It doesn't have to be a full-fledged, clear production, as we know from individuals with autism who are minimally verbal, but there is consistency in the intention of how they use that sound. So if you look at my cursor, you'll see that this is basically what we call the speech apparatus. So typically our tongue, our lips, our oral cavity, that's the articulatory apparatus. It combines with the phonatory apparatus, and that is the vocal folds, the larynx and the pharynx right here. These folds have to go back and forth, and the air goes through it in order to produce these sounds. But the air has to have a source, and the source are the lungs. And those lungs are what provide, through the contraction expansion, the puff of air that then we manipulate with this apparatus to produce the sounds we want. That is the motor act. But as we learn, and we know from child language development, that these very sounds eventually have to get mapped onto meaning. And we wonder, how does that actually happen? It's one thing to be listening, it's another thing to be producing it. And somewhere the two have to come together. And the whole process of extracting from the speech stream, from the acoustics, the very motor speech act allows a child to imitate. So when you think a child is imitating, what do you think they're doing? They've heard a sound, they're playing with their articulatory apparatus, and they're producing that sound. They're getting the mapping right by repeatedly trying and practicing. I didn't mean to ignore this part of the figure. This is also the part of the figure that's very important that we talk about in autism, because it talks about emotive content. So when we're trying to communicate, we use not just with speech, our happiness, our joy, our anger, our sadness. All the muscles from the face also come into play, in addition to the muscles of the lips and the tongue, etc. So if we were to look at this, what I'm presenting here, you're basically, and I'm going to pull all of this up at the same time. The sounds that Evan was referring to and spectrograms, you can think of similarly with the human utterance. In this case, the word is shop, and you can see it is divided into the sh or h. And we can actually classify and categorize these sounds with our equipment and place these cursors and break down a recording into its corresponding syllables. And once again, this whole act is made possible through the combination and coordination of these three systems: articulatory, phonatory, and respiratory. And I think a key take-home message, if anything, I'd like people to go home with, it's that perceptual production link. And this particular statement is really key, that when we speak, when we say the word shop, we are actually, these acoustics are bleeding into each other. So the p becomes part of the O, and the sh becomes part of the O. And yet our ears are able to segment this. And I think the statement here that in the acoustic signal lies the neurobiology of our articulatory apparatus. And that's what we, and in this project, are trying to do, extract these sounds and why something is going wrong in individuals with autism, particularly those who are minimally verbal, who have difficulty with production. And maybe more comprehension and etc. But we'll get to that. So really quickly, if we think about it, what is this project, which is a communication project, about? And here we're focusing on the speech component, though there's much more to it, which I will show you in the next slide. A quick way to think about speech when you're talking to your child or a family member, you can think of the form, which is a word form. That is all the acoustics and phonetics that make it up. You can think of content, which is the meaning of what the person is trying to say. And you think of it as pragmatics, or use, is how are they using that, though that's their speech, to convey meaning. Another way to think of the breakdown that might be occurring in ASD or in speech in a disordered population, is it happening at the level of planning? So when you say you're thinking of the word baby or apple and you want to say it, you have that word in your head, you plan the utterance ahead of time with your articulatory apparatus, you then coordinate all those gestures, you sequence them, and believe it or not, all this has already happened by the time you open your mouth and you say that word. And you're saying, how on earth did the child learn to speak? How did they ever learn to speak when they have to do all this stuff? And you ask yourself, where do you have evidence for this kind of stuff happening? So I can give you a very simple example. You've all heard of something called slips of the tongue, right? You can say, I had a, say, big dog, and someone might say accidentally, dig bog, right? You hear these all the time, and you wonder, how did that happen? The only way that could have happened is if the speaker actually had big and dog already in their heads, already planned, and like little slots, they filled these slots with these sounds. And just when they're ready to execute, something gets messed up in this process. And that's what this project is about, is trying to figure out where things are going wrong in this process. And another way is at a very basic level, like the big dog example. You can talk about the place of articulation. If b becomes a p or a d, the manner of articulation and voicing. So voice versus voiceless, or fricatives versus affricates. Place being going from the front of the mouth, like a bilabial, to the back of the mouth. Excuse me. So there is a very strong need for empirical research, and I'll cut to the chase here. Basically, we're trying to use a multimodal approach of developing a platform to get at objective measurements of sensory and cognitive problems that we can then develop biomarkers which become targets for our intervention. And we know that standardized measures, however good they are, with a population that has challenges like the population we are using and studying here, minimally verbal, the scores are not adapted for use with this population, and it raises concerns about reliability. And another simpler way to say it is like taking a square peg and putting it in a round hole. I remember this from my early research days when I was doing a lot of child studies, and my colleagues would give me all these tasks that were designed for adults. And then we'd give it to a kid and the kid couldn't do it. And then they'd say, oh, the kid failed. No, the kid didn't fail. You failed to design the task sensitively and appropriately for the child's ability. And that's what this study is all about, is trying to design tasks that our minimally verbal subjects with autism can do, and to whatever degree they can do it, to analyze their output. So this is just a quick schematic of the full scale of the project. This is the collaboration that we're talking about with the Lincoln Lab and MIT and Media Lab. This is what I will be focusing on now real quickly. The speech tasks are imitation, naming, reading, and spontaneous speech. And what I will be talking about is on the acoustics of it and the production aspect of it with EMG. We also have a number of other tasks, which are eye movements and hand movements and attention, but that'll be for another day. On the right, what you see is all the quick wearables that are used as part of the study, which makes it even more exciting and easy to some extent to think of the long-term implications for studying intervention and designing intervention. So real quickly, this is what a sample data set might look like. So on the top right, you see the person's face, and you see these little dots correspond to what we call little electrodes that are placed on the mouth, face, and neck, and they capture muscle movements, which is the electromyogram, EMG. On the top of the speech is basically the acoustics, so the sound recording. The bottom are the actual, each of these waves here. So each of these is a word that the subject named when presented a picture. So for example, they have apple, they have pig, they have dog and potato and so on. And each of the colors in here correspond to the different sensors and how they come together. And so to give you a clearer example, here on the left, you have individuals with autism, 2 samples, and here on the right are the control subjects. And what are we trying to show here? If you see this axis here, basically there's an x-axis and a y-axis. This is the time, and this is the amplitude of the muscle motor response. And you can see this is just before they start speaking, the whole anticipatory phase, how you begin to speak, and you can see the difference between 2 individuals with autism. And over here, in comparison, across the 2 columns are the individuals who are control subjects. So this is just to give you a sense of how we go about measuring these different characteristics of speech. Another example here, now moving from production to actually the acoustics, that is, instead of looking at the muscle movement, we're analyzing the speech acoustics. And a very common task that's used clinically is called the diadokinetic rate. In a simple sense, it's called a speech repetition task, and or a syllable repetition, and the syllables are pa, ta, and ka. So what you see here, the top is the control subject, and the lower row is the ASD subject, a sample. And here the subject is saying pataka, 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 pataka for a limited period of time. And the time is provided to them as a 1 second period or 1 minute. And so similarly over here, you have the individual with autism also producing the same sounds. And right away, just by eyeballing it, you need no expertise. There's no rocket science over here. You can see the big difference when you just eyeball these 2 data sets. And you can see that there is definitely, while there is a larger burst of strong sound, in that period of time, they begin to lose the energy and the respiration and the air that comes out that allows them to produce these repeated syllables. Whereas the control subject just chugs along happily and much faster. Over here also you can see it's like patika, patika, patika. It's a very labored, effortful production. And for those of you who are familiar with the concept of apraxia of speech, this is actually representative of a task that's used to diagnose this disorder in speech. So some quick take-home messages. One is that what we are able to see, even with the small data set we have so far and with incomplete analyses, and what I mean by that is that there's a lot we're still doing and need to do. We can see differences, which you saw in the diadokinetic task and in the various EMG waves, that there's a difference in coordination of the articulatory system. There's a difference in the loudness level, in the amplitude, in the way they can hold their breath and produce an utterance or a string of utterances. They even differ in the duration of a single utterance, and they differ in their loudness of it. The take-home message from this screen here, this message, a series of bullets here, is just this, that the whole motor speech apparatus is coming into play here and having difficulties in working together cooperatively in an efficient and accurate way to produce the sounds. And the struggle and the breakdown can be at many levels, and that's what we're after. So our translational goal, this project is not just about characterizing. We're very much interested in taking what we find in the laboratory and taking it out into the real world at home. And that's why our focus has been so much on wearables. And a big part of this next phase of this project is designing the feedback, the intervention. And what do we mean by that? Is that right now I showed you examples of production, EMG, and recordings of their speech. If our metrics are as good as I claim that they will be, those very metrics will become our biomarkers for targeted intervention. So we know that when we give feedback, what should we be enhancing? When should it be delivered? And how can we record that the effects of that feedback to be confident that we've really got our act together here, that the sensory and the cognitive parts are really going to be enhanced by this feedback, that this individual, that our kids with autism, our adults with autism who are minimally verbal, it's like almost that they're suddenly becoming very aware of their selves, their systems, and they're able to produce that output that enhances their communication, because it's all related. The social ability is related to our ability to speak, and vice versa. So, and what do I mean by that? When kids have a very large vocabulary, they get better at speaking as well, because that large repertoire massages those very ingredients that are impaired at this stage. So it's a win-win situation. When you improve vocabulary and language, you're improving speech and vice versa. And with that, I'd like to end and thank you for your patience and spending some of your evening with us.
Fantastic presentation, Maria. You can go ahead and end your screen share, and we will go ahead and jump into our discussion section. Many of you have already sent in some excellent questions, and I've got those queued up here to pass back to our presenters. But as we're going through these first questions, if you have other questions for Maria, particularly if she got through the last portion of her talk, feel free to send those in the chat or hold on to it, and we'll save some time for folks to unmute at the end. We're going to start by popping back over to Evan. And Evan, I'm going to throw a couple questions at you that were a little bit related. So we had some great questions really about sort of the beginning of all of this rodent research in autism. How did you and others before you come up with these mouse models for autism? And why did you think about researching mice instead of other animals whose sounds we can hear, right?
Sure. So I think that one thing that I'm just going to start by reiterating this, and I think a lot of people don't always make this painfully clear, but there is no such thing as a mouse with autism. That is very much a human condition. What we're doing when we're creating what we call these mouse models of autism we're trying to create a mouse that has some type of validity for something that we see in humans with ASD. So, one thing that we know there are, of course, certain genetic mutations or deletions or something changed with different aspects of genetics that we can correlate very well with someone who may have that particular form of autism, and we can knock out, duplicate, alter that particular gene in a mouse and look what happens. And oftentimes we can see very similar changes when we look at a more, biological molecular level. We can see the same kind of changes in the blood in certain cell types as we see in people who are diagnosed with autism, we're seeing in these mice. Or we can take epidemiological data where there's a very strong increase in children diagnosed with autism when their mother had a particular type of infection at a particular time during pregnancy or something similar to that. And we can essentially model those types of things and then look at the effects on different behaviors in mice, different molecular or biological changes that we see in the mice. So we're not giving our mice autism because that's not a thing we can do, but we can model certain aspects of it in the mouse and then use all of the advantages that we have in mice, the ability to manipulate different things, the ability to directly manipulate things, directly access tissues. And we use mice really for those reasons. And the main reason that we currently are using mice rather than, some other animal species that you can, you don't have to go to all the pains of getting specific expensive ultrasonic microphones, doing all these different arrays that we have to do, are mainly that the scientific tools right now to manipulate genes are much more readily accessible in mice and rats than they are in any other animal. So in all honesty, it's just, they're much easier to use and they're kind of where the science is at right now.
It's clear in your answers, Evan, that this is all sort of a journey and process. And I imagine 10 years from now in your career, you'll be looking back at some changes in how we have modeled autism. As you think about that, what sorts of improvements do you think we as a team or, you and your other basic science researchers need to work on to improve our ability to research some of the phenomenon of autism in rodents? Where are the biggest bang for the buck? If we could just do X, right? You talked a little bit about that open social model, which might be one of those that you're working towards. But where do you think we need to take the measurement in your research?
Yeah, I think that honestly, what we do here at the Lurie Center is exactly where we need to take this. And it is clinicians, parents talking with basic science researchers. We can kind of be the paradigm of what you think of when you think of a scientist. We can be walled off, hanging out with our mice, not talking to anyone, and really kind of live in our own world. And I think definitely when you do interact with a lot of other scientists, you see that, and they're not really thinking about how do these measures matter. And I think one thing that is sometimes scary when I'm just going to use the vocalization behaviors that we were talking about today as an example, and you'll see some scientists will publish a paper saying that, hey, look, we have a mouse model of autism because we actually were able to increase the number of times that a mouse made a vocalization. What does that mean? And, I think we have to look at it from a lot more complex manner, and we have to really look at, of course, we don't yet know what any of these little squeaks or spectrograms are meaning, but we can look at how they're changed and not just say, hey, there's a change in the number, but just kind of like everything that Maria was just showing, what goes into the structure of these vocalizations. And I think we're going to learn a lot more by doing these more complex measures and these more complex assays.
That's great. Really helpful. I think on that note, we're going to go ahead and jump over to Maria for some questions, but I've got a few more for Evan, so we'll come back to you if we have time as well. Maria, as you've heard some of Evan's responses, tell us a little bit about your thoughts on how we might be able to use some of this basic science data and sort of these mouse models of communication and autism to help autistic individuals, right? Which is really at the core of the translational work that you're aiming to do.
Yeah. So I think mice might be a little easier to work with. Forgive me, Evan, for saying that, but you probably know better than I'm not in the trenches with you there. But trying to get compliance sometimes is hard, and therefore we might not be as accurate as we'd like to be as researchers with our human vocalizations and getting them under conditions that we would like to be able to be say with confidence that what behavior they're associated with. Because one of the things I always have difficulty with is putting our subjects, our participants, our kids, our adults in these very confined chamber-like settings. And not that mice aren't being put that way, they are, but I think that you can let a mouse run free, you won't have, and I think you could still get pretty good data. I think if we let our subjects run free, we won't get any data because they'll run away from us. And so I think the idea there is very, I think, interesting to do that connection with what Evan is doing, because aside from the jokes, my half-hearted attempt at jokes, is the fact that and I'm not sure, Evan, you can correct me about this. You know, the knockout mouse models, one of the things we learn about autism all the time is the heterogeneity of autism. It's a polygenic disorder, right? It's very hard to be certain that we're talking about the same autism. You know, it's like there are just so many subtypes. And we just don't even know when in the whole process things are breaking down and how they're breaking down genetically. I mean, we talk about environmental issues. We talk about prenatal and perinatal and postnatal insult. There are so many things, but genetically, the way some researchers have tried to console me as a person is saying, oh, Maria, why don't you just study the monogenic form? You know, you have a single gene, you knock out that gene, and then you go. And I say, but there goes your generalizability. And that's what we're up against as researchers in trying to do translational medicine. So to answer your question, Lisa, and trying to bridge that gap, I think you can get a lot of behaviors, and you can actually do some more interesting paradigms with mice, but I'm not sure the interpretability would necessarily translate as easily as a result. So that's where it's a double-edged sword.
Yeah, absolutely. I think your response there, Maria, does highlight this real variability in individuals with ASD. And, you talked a little bit about some of those complex factors, including, the sort of speech generation process where we're looking at planning, coordination, sequencing, that all has to happen before execution happens. In addition to those sorts of variables, are there other reasons why people with autism may have so much variability in their vocalizations and communication? And we have a related question about Gestalt language processing. So kids who might be identified as Gestalt language processors, what are some of the other reasons why you think there's so much variability in how individuals with autism speak and communicate?
There's variability because I honestly think there is just so many types of autism maybe, and so many characteristics of neurobiologically different. What was the trigger that caused somebody who have a child with autism? Is it regressive? Is it a type, there are things that we can separate by saying someone who comes to me and say, oh, my child used to speak and doesn't speak anymore. Or someone, or for example, my child was fine and suddenly had a really bad sore throat or a cold, believe it or not, something as simple as that, very high fever, and they lost a lot of their social skills and with that verbal skills. And that became very interesting to me because I started asking the question about the interaction between social skills and linguistic ability. And one thing we know, and even Evan's work alludes to this, is that these creatures are, just by virtue of being who we are very social. We are born to be social. And you see that right from the crib, whether you're talking about an infant or you're talking about a mouse, or you're talking about the mother of a mouse or the mother of a human child, there is constant interaction. And when that interaction is in any way impaired because they're not getting responsiveness or something in their system is not responding the way it is, the answer to that question would be they're not getting the necessary reinforcement, reward, and feedback that's part of the loop that allows us to continually build and develop more. When you ask what a child at 18 months to 5 years, yeah. And then your second question about the what did you call it? Gestalt.
Gestalt language processing.
So I would be a little worried about using an approach. This is my opinion. It doesn't necessarily represent the opinion of others. I'll be very clear there. To try and just focus on getting a kind of a whole language approach or a gestalt approach, if that's what the person who asked the question is about, because getting the units of language is very important in order to develop the combinatorial abilities. And so whether it goes from speech to reading or vice versa, reading and speech are integrated like this. And the one can help the other. So please don't just use gestalt. Go with the smaller units, go with the syllables, go with the sounds, play games, and develop and massage their whole sound system.
Thanks, Maria. That's really helpful food for thought. I think your response here makes me think about this idea that folks have said many times before, that when you know one person with autism, you know one person with autism, right? There are so many factors that you've alluded to that can contribute to that variability. Yeah. I want to pop back over to Evan for just one last question. We're coming right up at the hour here, but you guys had so many good questions. And there really are two, Evan. So I'm going to give them both to you, and you can either pick what you want or answer both if you think you have time. But in your analysis of ultrasonic vocalizations, do you ever see mice that are sort of nonverbal or non-speaking that sort of present a proxy for this population we see in a certain percentage of autistic individuals? And then related to that, has there been any research done on postnatal immune compromise? So you talked about sort of those prenatal hits, but anything on postnatal hits? So two different questions. You can pick which one you prefer.
Sure. So yes, we see plenty of variability in our mice. It's always kind of shocking when you think of, we use these inbred mice that are supposed to be the exact same thing. Every single male mouse is supposed to be the exact same mouse. It's not true at all. They have incredible variability in things such as their vocalizations. There are some that are essentially chirping away all the time, and there's some that we see that just make, next to no noise, or the only thing is an occasional little squeak that very clearly probably doesn't mean all that much. And we definitely think of it very much in terms of, like you said, the minimally verbal. And then, yeah, so we actually do see with these kind of, postnatal immune challenges, give a mouse a fever and there are alterations in their vocalization behaviors very similar to what we would potentially see in the human condition. Obviously it's different, but we do see differences with these kind of postnatal immune conditions.
Lots to learn from you and your teams both. Thank you both for your excellent presentations tonight. And thank you for all of our audience members for coming. You had such excellent questions. I so appreciate your engagement at the end of a long day, at the end of a long summer. So thanks for joining us. And we hope that you'll be back in September when we talk a little bit about a new IEP form in our next month's Coffee Convo. If you haven't heard, the state is coming out with a new IEP form. So plant that seed. We'd love to see you back in September.